I'm Robert Henry from the Centre for Crop Science, and I'm going to chair the event today. I'd like to start by acknowledging the traditional owners and their custodianship of the lands on which we meet today. I pay respects to their ancestors and descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australia and global society. Just a few housekeeping things to mention. The seminar is going to go from 12 noon till 1 p.m. At the conclusion of the seminar, we will hold a Q&A. If you've got questions online, please use the Q&A button rather than the chat button at the bottom, and we'll address them at the conclusion of the seminar. So it's my great pleasure today to introduce to you Professor Bob Gilbert. And I've got some words written here that I'll read that have been prepared so that I don't stray from the script. Bob Gilbert did his education at the University of Sydney and Australian National University and MIT. He worked at the University of Sydney for many years and moved to UQ in 2006 to join the new group, which became the Centre for Nutrition and Food Science. He's written over 500 papers and 2 books and been an advisor to more than 100 younger scientists. His research achievements can be best described as developing theoretical and experimental tools which can be used to obtain new knowledge and insights in apparently complex systems. It then says I can add anything personal I'd like, and I might just refrain from that, Bob, and ask Bob to come up and give the presentation. I've been failing to go through my slides. So we've done acknowledgement to country, and that's the housekeeping, and it's now over to Bob.
Robert, for telling such kind lies, and I will pay you afterwards. Okay. So, now, here, and Yangzhou in China, and this is, well, the tale of 2 molecules that we eat one and make the other all day and a fair bit of the night. They're very important for our health. Now, I've tried to name most of the people. I've probably missed out on 20 or 30. Forgive me. I will you tell me about it and I'll wear barbed wire underwear for as long as you wish. Now, I'm not going to try to name everybody. Goodness. Robert, I don't think you're on there. You should be. Oh dear. And these are the people who really did the work. Now, what am I doing here? Now, I said for about the last 8 or so years, 9, I've lived in China half the year. And 6 months in each place. And, whoa, what have I done? Yeah. And at Yangzhou University, which is a relatively small university, but it's very good in agriculture in Jiangsu province. Now, so I'm really like that. Now, these are going to be, this talk is going to be about two very similar molecules, both of which are complex, branched glucose polymers. And starch is what plants make for their energy reservoir. We eat the plants, and that then becomes, we convert it to a very similar molecule, glycogen, the same bonding, it differs at a higher level, which is our energy reservoir. And starch is roughly half our food energy. Let's do a little bit of history. Over, let's say, the last 5 million years, in fact, since I was a young man, there's been our digestive system has optimized to our diet as it was for most of the 5 million years. And these were coarse grains and tubers, the occasional other human being. And all of these are hard to break down to glucose. Glucose is then what provides our energy. So what we have developed is getting used to food that takes a long time to break down to the molecule that we want, glucose, which drives everything. That's okay. Fine for our insulin system. And there's also a little bit of starch that gets down to about there, resistant starch, which our gut bacteria are saying, oh, thank you, lunch. And they then produce things which help fight some of the bad products formed when we eat red meat. Red meat's a perfectly natural part of our diet. So if we become vegetarians, there's nothing wrong with that, but we have to balance the amino acids. So that's what we evolved. Because red meat, or the byproducts, cause some DNA damage. But digestion of things, we make some short chain fatty acids, butanoic acid, butyric acid, which help overcome that problem. So all this involves our evolution for a body that does its best to overcome the problems of obesity, don't say a thing, diabetes, and colorectal cancers. Then we made a really big mistake. We invented agriculture. Robert, what does the first A in coffee stand for again? Because it enabled us to start producing a lot of our food without having to go and hunt or dig for it. Very efficient food production. And this freed us up to do things like write poetry, do science, and some other things that I won't mention. But then we, as a result of agriculture, changed to a diet that has a lot more starch in it. And that's not what we evolved for. So that's okay while we were busy working hard in the fields. And then we started leading sedentary lives over the last, well, few hundred years, depending on which country. And then the things that, all that starch goes now to fat, and so on and so on. And that is bad for us because we've so much changed our lifestyle. Now, at the moment, I don't know if you can see that, your lunch is turning too rapidly into this. We have obesity and colorectal cancers and diabetes in big numbers. You can argue about glycemic index. I don't want to get into that. But what is important, what has been found, is that so-called resistant starch, starch that lasts down to about there, is actually good for us. It gives lower levels of obesity, colorectal cancer, and diabetes. Now, in countries like Australia and now China, those are becoming epidemic diseases. They're at the epidemic level. And it's because of our change in lifestyle, the food, and being more sedentary. So, as I said, it's becoming bad in China. It's a major public health problem. By the way, can you hear me at the back? Is the microphone working? Yeah, thank you. As long as the thumb is there and it's not one of the other fingers, that's fine. So what we do is we'll eat starch that goes into our little tum-tums, gets digested there to glucose, and we then store that in a very similar molecule, glycogen, if you're a mammal. I think, yes, everyone in this room is a mammal. So starch is this highly branched glucose polymer. The branching can be arranged either a few long chains, which is amylose, or lots of little short chains, which is amylopectin. And you have both, except in some unusual cases. And it has a complex multi-level structure. I won't go into it, but you can actually identify 5 different structural levels for this molecule, which is what attracted me to this in the first place. I started hearing about it when I was at the University of Sydney, thinking, you know, the field that I was then in, emulsion polymerization, which I turned into some nice mathematics. And I was thinking, okay, what do I do now? Then I started hearing people, including Mike, talk about starch and this complexity, and there was no mathematics associated with it. Now, I've got a problem, a very severe problem, and I'm to be pitied. I like mathematics. And I don't think there's anything that can be done about it. So how could I mathematize this? By the way, diabetes is bad, including in China. So with this complex structure, what are the features that control digestibility? There are the 5 structural levels, sometimes you can identify 6. What is it about these? Now, diabetes starts with glucose spikes when our food is rapidly digested, and different types of starch are digested at different rates. So if you have food where the starch, nothing much happens to it, it sits about there for a long time, that's really good for you because you then have slow glucose release. You don't get a glucose spike. Oh, something I forgot, forgive me. I realized, look, everyone is used to doing things by Zoom. So just to make people feel a little bit more comfortable. Okay, is that better? Yeah. So, what was in there? You see any strange creatures crawling on? Okay. So food that's slowly digested is good. And that's controlled by structure. Now, how do we measure molecular structure? When you've got these multiple structural levels. And, well, there's one which is relatively easy. You've got all these branches. Remember, it's highly branched. You add a debranching enzyme. You then have all these branches. You then measure the distribution of branch lengths, and that's doing it using a machine called FACE, or sometimes that bloody machine, or Fluorophore-assisted carbohydrate electrophoresis. That's one way of doing it, at least for the shorter chains, namely amylopectin. Longer chains, you have to use a technique called size exclusion chromatography. We get out how many chains have which length. SEC separates by size and not molecular weight. If, sometimes called GPC, well, that's not strictly correct now. If you read in a book that says SEC separates by molecular weight, burn the book. It's wrong. It separates by molecular size. If it's a straight chain, that's fine, because then chain length and molecular weight have a one-to-one relation. But if it's highly branched, like starch, no. So this is the other technique, Fluorophore-assisted carbohydrate electrophoresis. And we have two nice machines in China, zero here. But basically, I won't go into how it works. That's how it works. And you get individual chain lengths coming out. It'll only go up to DP. Well, the highest we've got is about 150. Okay, we get out these plots. Oh, good. What do we do with them? Because at the moment, they're just pretty pictures. Okay, we need to learn something from these pretty pictures. So we need to be able to reduce these data to something meaningful. And that's where some knowledge of the biosynthetic processes and the mathematics comes in. When I first got into this, started hearing about this, I was told by somebody known to many here, Melissa Fitzgerald, look, Bob, it's too complicated. You'd never be able to reduce it to the sort of mathematics that you like to do. It's too hard. Well, okay, Melissa was wrong. It took quite a few years, but we did. Oh, it's complicated though, because we get all these data. Well, what are we going to do with all these data? The very important thing is to say, wait a minute, let me try to find the important thing in these data. This is a painting by the 16th or 15th century Flemish painter, Bruegel, sorry, my Flemish is not what it should be. And it's called The Fall of Icarus. Icarus, you may know, Greek legend, a young, brash chap who knew he knew more than his master, so he put wings on his back, glued them on with wax, because he wanted to fly, and he started flying, and he flew so high that he got too close to the sun, and the sun melted the wax, the wings came off, and bye-bye Icarus. He fell to his death in the ocean. This painting by Bruegel is called The Fall of Icarus. Huh? Why is it called that? Here it is, a scene of late medieval life. Why is it called The Fall of Icarus? You've got to look for the important thing. there's the important thing. It's almost hidden. You have to look hard to see what is important. Okay. All these gazillions of enzymes that are involved in all these different processes. Oh, it's too hard. However, the important thing is that there are only 3 types of enzymes. And on good days, I can count up to 3. So these enzymes, starch synthase, which adds glucose monomer units. There's starch branching enzyme, which forms branches in a rather complex way that I won't go into, and starch debranching enzyme, which completely removes branches. Lots of enzymes, but they're all types of these. And then, oh joy, oh bliss, oh rapture, you can write down the maths. And that's something Alex Wu did. In fact, in his honours year, wrote down the maths, solved the equations. If you like matrix algebra and eigenvalues and all that sort of stuff, it's really nice. Again. So we also, so that's for the amylopectin. We've also developed, and that's by Sharif, wherever Sharif, oh, he's not here. He's writing a thesis, so he's probably having a nervous breakdown at the moment and biting bits out of the carpet. If you want to know where Sharif is, look for bite marks in the carpet, you'll find him. So we've developed models for amylopectin biosynthesis and amylose biosynthesis. Okay, it does involve mathematics. It involves multiple sets of enzymes, but there are only 3 sorts, as I said: starch synthase, starch branching enzyme, starch debranching enzyme. And when you really think about it, for each set of enzymes, I have lots of different sets of these 3, but you only need 2 parameters for each set. One turns out to be the ratio of the activity of starch branching enzyme to that of starch synthase, a different one for each set of enzymes, and the relative rate at which that starch synthase propagates, beta and H. So it's only 2 per enzyme set. And you have the math. Oops, I thought I was speaking to Americans now. I'll get back to English. Well, you have the maths, and you can then fit it to the 2 parameters, except you might have quite a few of those 2 parameters. And we've written code to do this. We happily make these available to people. And quite a few people in China now come to us because although we write instructions, it's not that easy. You really have to have somebody holding your hot little hand. And they accurately reproduce the data when you fit it. Well, let's look at some results. As I said, one of the things that you can do is add a debranching enzyme. What is the weight of chains that are this big or have a certain degree of polymerization? And what you see is that there are clearly multiple contributions, but with the maths, we fit these. And what we end up with is being able to reproduce these data with a small number of parameters. Now I'm going to give you an example. This was done by Hong Yan, who did his PhD here. And basically, oops, if you have things like spatial, try that again, distribution from FACE or from SEC, you can fit those and you get a small number of parameters. If you have a small number of parameters, then you can start to use those to find correlations and thus learn about what the system is doing. And what property might we be interested in? One is sensory. Another is digestion. How good does it taste? And is it digested quickly or slowly? So here's how you do sensory data. You have a panel and you get it. You can quantify it. Many thanks to, I'm not sure if Sangeeta's here, but we've done this with Sangeeta here, and we also do it in Youngjaw. And we can quantify sensory into a small number of parameters. And there's just one little problem. Slow digestion, good. Rices that are slowly digested taste bad. Problem. Because people, no matter how good it is, they don't like eating something that tastes bad. Well, that's a problem. And it's a problem that we are, I'll just go through I won't go into the sensory in detail. A lot of data on this. We get data, we get parameters out. We can understand sensory in terms of the molecular structure. And you can have, let's take rices that are slowly digested, but they taste bad. Now, what we've found, and this is some important work done by Jifwe here, is that the molecular structural features which control the sensory are not the same as those which control digestion. So they're different parts of chain length distributions. And that way, you can now, will be able to, because it's something that Jifwe is working hard on. You don't sleep, do you, Jifwe? It's very bad, very, very, very bad. That way, they're actually two different dials you can control. So it is possible to make things which will be both slowly digested and have acceptable taste. We're not there yet, but we shall get there. That's just saying that. Oh, it's thought that, oh, it's just a matter of how much amylose, but it's not. That's a major determinant, but there are other ones. Okay, now we're going to change tack now, change direction, sort of, because we're going to look at, we've looked at starch, one glucose polymer with a complex branch structure. Now, we're going to look at another one, glycogen. Now, if it's a, starch has this highly ordered structure, at least part of it, highly ordered, but glycogen is quite random in its structure. Range of molecular weights. Now, right now, if you had lunch over the last hour or so, you're converting starch to glycogen. Because now you have another glucose polymer, and that's, you store it until you need it, until you need the energy. Now, we're now going to go to diabetes, because this is getting to be a very bad problem. And I think the statistics are, in the next 30 years, a third of the population in countries like Australia is going to have diabetes unless we do something about it. And that's a debilitating disease. And I won't do it today, but sometimes I've asked people to put up their hands if they or a close family member has diabetes. Well, that's a lot of people, you find out. It's becoming frighteningly common. And it's becoming worse and worse in China. And that's going to be the world's biggest public health problem over the next 30 years, the high diabetes rate in China. Okay, glycogen is where we store it. And it's this random structure. Instead of the sort of ordered structure that you get in starch, you have, well, you can identify 3 levels. The individual branches, then they're joined together in the whole molecule. And then what happens is those whole molecules themselves join up. It looks like a raspberry. They join up to form, well, the little ones are called beta particles. When they join up, they're called alpha particles. And after you've eaten, and perhaps an hour or so later, you're getting lots of alpha particles, which are then more slowly broken down. So that enables you to control your glucose level instead of getting a big rush of glucose and ending up with diabetes. So, as it said, it has these 3 structural levels, and the same as in starch, you've got the alpha 1,4 linear links and the alpha 1,6 branch points, and beta, and then alpha particles. The biggest reserve for us is in the liver, but it varies throughout the day. As I said, we've got diabetes epidemics in the US and Australia, and even higher levels in China. There's an article which is almost 10 years old now, pointing this out by some Chinese authors. And it's a big, big problem. Well, studying this is a bit difficult because starch, you've got a molecule and you have it in plants, and we don't worry about cruelty. We don't have ethical requirements for plants, but we do for people. We actually do some human, I won't say experiments, we have to be very careful, but with full ethical approval, we can do certain limited types of experiments with humans. Much easier to get the ethical approval, fortunately, in China. There's no danger, and it's easier to get approval to do the same thing in China, which is where we do this now. So, and of course, you can do it in mice, and you can have ordinary mice and mice that have been engineered to not have the satiety gene. They keep on eating. Sometimes they're called American mice. And are there any Americans here? I'll explain it to you afterwards if so. So the DBDB mice, as they're called, that's the animal model. We use the same equipment as we do for starch. I won't go into these. Oh, what have I done? Because many of these transparencies are also used when I talk in China, so you also have English and Chinese, and if any of you who are Chinese find any errors, please let me know. It's not my fault, it's somebody else who translated it for me. Now, it's not easy to get the glycogen out, and some of you know, very hard without, well, it's easy to get it out, but without degrading its structure. Developing MENS, and recently D has got some very, very good results for this in working in Suzhou and in Wuhan. And we, again, look at size distributions, weight distributions. Well, there's glycogen alpha particles. Now, we made a discovery probably about 8 years ago now you have the little ones called beta particles. They join up into the big composite particles called alpha particles. Well, what we found a number of years ago is that if you do things like adding dimethyl sulfoxide, then what happens is the big glycogen molecule can break up into the little ones. The alpha particles can break up into the beta ones. If the glycogen comes from diabetic livers, and much less so if it's from healthy livers. So it's quite a spectacular difference. The disintegration of alpha particles we think it's because of hydrogen bonding effects, although not quite that simple. Some very nice experiments that Sindler has been doing. It's not any old hydrogen bond breaker that'll do it. But basically, you have diabetes, your glycogen falls apart much more easily in DMSO. Now, obviously DMSO is rather poisonous. That's not clinically relevant, but it says there's a difference in the bonding. So we don't know what that difference is, and we are trying to find out. Some very nice work being done by Sun Liu in our group in China. He's also been doing it in Suzhou, trying to find out just what that bond is. But it goes wrong in diabetes. Finding out what that is going to be a big breakthrough. If we could do that, we'd have new drugs for diabetes management. Now, how do we know these things? Okay, we take healthy mice, we sacrifice them, extract the glycogen from the liver by a method that doesn't break it up, and look at its size distribution using size exclusion chromatography. And the blue is the native, and the red is after you've added dimethyl sulfoxide, which breaks hydrogen bonds. Healthy, no change. Diabetic, big change. Diabetic, what you see is that the glycogen breaks up into those component particles if you add a hydrogen bond disruptor. There is therefore a difference in the bonding. Now, we're trying to find out what that is. It may be that the molecule that ties them together may be a protein called glycogen, but there's evidence both for and against that. And Sindler has done some very good work on that. Unfortunately, we don't have the answer yet. We're trying to find out. But you can obviously see that, wow, if we could work out what is making that difference, and is that difference clinically relevant? If that's happening inside our bodies, if you've got a big glycogen molecule, enzymes are doing their work on the surface, slow degradation to glucose. Lots of little particles, lots of surface area. Those same enzymes are going to break them down to sugar, to glucose much more quickly. Diabetes is uncontrolled glucose release. So clearly, this result is clinically relevant. We don't know how yet. But again, we're starting to be able to, with full ethical approval, do some experiments on people. We can do them fairly easily on mice. That just says what I've said, that diabetic alpha particles break up more easily than do healthy ones. Don't know why. But a little part, if that happens in the human body, and obviously not with dimethyl sulfoxide, if that happens in the human body, then that's highly relevant to the loss of blood sugar control that is diabetes. That just says that with nice, well, not nice, but pictures. Also, we do the same trick we did with starch. We add debranching enzyme and look at the chain length distributions. And look, what you see, healthy versus in blue, and diabetic in red, there's a big difference in the chain length distribution. What you have is more longer chains than the diabetic. We don't know what the health implications for this are, but we know how to control that because we know what enzymes control chain length. In fact, very similar to the enzymes that control chain length in plants. Very similar enzymes because they have the same function. And there are drugs which can then control activities of these. And goodness golly, gee whiz, we found that one of the drugs that is used to help manage diabetes also controls the chain length. It was discovered, and that's probably why it works. So now, working with our friends in pharmacy, in Yangzhou and in Renmin Hospital, both obviously in China. Okay, how can we start to use that knowledge clinically? New drug design. So, oh, good heavens above. There's also a difference in molecular density between healthy and diabetic. I won't go into that. That's when you use a laser light scattering detector. What about humans? These have all been done on poor, unfortunate mice. But we've also been able to do this with humans. And without going into the details, we have friends in hospital who, with full patient approval and full ethical approval, this is all done in China because that's where our friends who are high levels in hospital work. And they can then ask patients, would you mind giving us, we have to do something to your liver. You have to have liver surgery. Can we take a gram, an extra gram? And we indeed see some of the same things in humans. Obviously, it's much more limited because as soon as you start doing surgery, of course, the patient has had to starve for 8 hours and that changes the glycogen size distribution, as we see in mice and rats. But we're again starting to be able to do this with human. And you see the same fragility because human patients who have to have liver surgery, some of whom are diabetic, some of whom are not, you see the same fragility in the diabetic ones. So it ain't just mice, it's people as well that show this. Well, what causes them to stick together? If you can tell me the right answer, I'll pay you $10 and share the Nobel Prize. They're very difficult experiments to do. Sinler has done some really good work, but it might be a protein. The problem is there's so much protein there, and there are probably only a few that do the binding. It's like trying to find a needle in a haystack. Proteomics is very, very difficult. But obviously, it's something on which we're working. That's all about proteomics. Why do these alpha particles form? It's clear that they're important for diabetes. Whoa, we'll be doing lots of things on that, and I'm not going to go into the details of what we're doing, but it's a complex cycle in humans and in rats, which we're starting to understand. And we also ask ourselves, well, what about the physiological things involved in these alpha particles? We're also working on that. And one thing, you've just eaten lunch. You're storing the glucose that you've digested into beta particles. Well, why doesn't it keep growing? We answered that question theoretically from simulations a year or two ago. When you do high-level molecular and dynamic simulations on this, you discover that it grows bigger and bigger, and it starts to get more and more dense. And then it just can't grow anymore. In the simulations, it gets so big and then stops growing. You're adding and breaking bonds all the time. It's a dynamic evolution, but the simulations, just adding your standard enzymes to the simulation, give you roughly the size distribution you see in experiment. We'd like to be able to control that, although people before thought it was due to crowding, but they're wrong. And those are the simulations, which I won't go into. Uses a lot of computer time, but it's actually hindrance, the opposite to what people had thought. And indeed, Queen Besford then did some lovely neutron scattering experiment, which proved this. It's opposite to what had been thought for 50 years. And we can start to see now, well, how you could start to make drug interventions as you understand the mechanisms, because you can change these activities pharmaceutically. And I won't go into the fragility of alpha particles, but basically what this is leading to, we aren't there yet, are new drug targets for diabetes. That weak bonding that you see in diabetes, well, what causes it? And if we knew what caused it, which particular enzymatic pathway is going wrong, that's where you can have drug interventions. So we're trying to do that, and we're trying to find out just what that bonding is. It's very hard. And Singler's been doing some fantastic work here at UQ. So I think that's everything. Yeah. Well, the well-known tribe in Africa, the Halawi tribe. The Halawi tribe, ladies and gentlemen, live in the grasslands in Africa, and they are 1 meter high. And the grass in which they live is 2 meters high. And the Halawi tribe in Africa go around saying, where the hell are we? Where the hell are we? Well, where the hell are we? We've found out a lot, and we've got a long, long way to go. I haven't told you about controlling chain length distributions in plants, which we can do with some clever GM techniques. Li Cheng did some really good work on that, and I think there's someone here who is also working on that. So we start to understand palatability, And you've got to compromise between good palatability and slow digestion, and that's something which is again being worked on here. And we're applying this sort of methodology to starch's cousin, glycogen. Okay, that's everything, yeah. So that's where we are. We have a long way to go. Fortunately, I'm only 75, so I've got a lot of time. Thank you very much.
Well, thank you very much. A very illuminating lot of work. We've got some questions, and we can take some from the floor and some online. I'm not sure whether we've got them. Do we have any here?
I wonder, should he talk into a microphone? Or? Yeah.
I have to read that.
Yeah, so my question is, you looked at the rate of converting glycogen to glucose. Did you look at the opposite direction? Like how was?
It has to be really people who've grown glycogen in vitro,
but it doesn't quite have the same structure. And the trouble is trying to do it well in mice. The thing is that it's on all the so
no. Yeah. I think I had a question, Bob. Concentration?
I think it is. I haven't gone into the experiments that have indicated this. Yeah. We think that's all it is. It's the higher blood glucose level. Oh yeah, you don't even need any mathematics, but some experiments that also, I always distrust maths.
I think that's been it's great to see that two parallel lines of work the parallel line first we got to that they were a fair way apart, bringing a lot of close to linking up. That's great to see. So I'd like you to join me. Thank you. I just our next science seminar is from Mo from the Centre for Horticultural Science, and that's on the 30th of November, and I think you get more details on the website. The other thing to note is that we're trying to get some feedback on the seminar series itself, and those of you that are online, I think there's 2 QR codes, and so it's the one on the right that allows you to give some feedback on the seminar series. The one on the other side, it just tells you about the seminar series. So it's the information and the feedback. So please make use of that to give us some feedback on the overall program. And that's all we have today. Thanks very much.